All right, everybody. So let's talk about lipids in a little more detail. So fats, what are they? What are they made of? These macromolecules are made from a couple of things. First off, they are made of glycerol, which is a three-carbon alcohol, and is also made of something called a fatty acid. All right. And these are also hydrocarbons, meaning, as you can see here, they are made of hydrogen, oxygen, and carbon, just like carbohydrates are. Same three molecules. As you'll see really quickly, these chains can be a lot longer. All right, but they're made of the same three components, same three elements. They are also put together, combined using the same process of dehydration synthesis, condensation reactions, same thing. And that's how you combine fats together. Here's an example of a triglyceride molecule. So, as mentioned, they are hydrocarbon chains, and they have very long skeletons. So they're very long. Okay, just like we talked about before with our glucose molecules. Glucose only had 6 carbons in it. Um, Fats typically have about 16 to 18 carbons per molecule, and these can be hundreds of molecules long, so they can get very long. That said, another really unique characteristic is they are nonpolar. All right, meaning that they are hydrophobic. They don't like water. If you don't believe me, take oil and put it in water. See what happens, right? And that's exactly it. They don't like each other, so they're going to create. They're going to separate from one another. You can see this in anything. You can just take oil from like your kitchen, toss it in some water, put it in a water bottle, shake it up, see what happens. They'll separate. And you can also see this in things like oil spills, and that's why the oil will float to the top of the water, and sometimes can be skimmed off as much as possible, at least. And that is the idea for the non-polar component. They do not mix with water. Um, that said, some characteristics, as mentioned, they are insoluble in water. The source of variation among them is their fatty acid composition, so that's what it depends on. Fatty acids in a fat may be all the same. Some can change. They can vary in length. They can vary in the number and location of carbons, and that's another important thing. It's not only just a number, but also the location of those carbon molecules. And the two that we typically talk about are our saturated fatty acids and our unsaturated fatty acids. We can go on and on about polyunsaturated, monounsaturated, and just keep going with that. But for this, we're just going to stick with saturated versus unsaturated. And if you're looking at this picture, what you're noticing is these double bonds. So in an unsaturated fatty acid, you're going to see these double bonds form and put a kink into the structure of the fatty acid. On top, it's more linear. Down here, you can see this little bend in the fatty acid. What is this going to do? Well, for starters, saturated fatty acids are going to have no double bonds. It's going to be a very long chain. There's going to be no real kinks into it. It's going to be a straight chain. And where are we going to find this? Typically, it's going to be solid at room temperature, and you're going to see this in animal fats like bacon, lard, butter, and so forth. Unsaturated fatty acids are going to be fats that have one or more double bonds within them. So one or more of these going on. When this happens, it's going to create a kink in the chain, um, and it's not going to allow them to get close enough to solidify. All right. So with that said, an example I use is imagine you have sticky notes, right? A stack of sticky notes. If you have a full stack of sticky notes, brand new, they're going to look more like saturated fatty acids. They're all going to be very compact. They're all going to be really close together, and it's going to be a solid structure, right? However, if you maybe you put them in your bag, and those sticky notes start to get, ruffled around a little bit, and they start to bend, and they get all crumpled up. It's not going to be this nice flat rectangle anymore. It's going to get big, and you see a whole bunch of gaps in it, and it's going to be a mess. In fatty acids, it's not a mess. It's just that these bonds and these molecules are not going to be super close together, which means they're going to be a little bit further apart. This is going to allow them to maintain a liquid state. at room temperature. So you're going to see this in a lot of plant fats. So corn, peanut, olive oil, and so on and so forth. So fats, as many of you know, is an energy source, and it's a very efficient one. So with that said, 1 gram of fat stores actually it's more than twice as much energy than that of a sugar. So in other words, if I had 1 gram of a sugar and 1 gram of a fat, a gram of fat will burn over twice as much calories as that of the sugar, even though they're the same weight, the same mass. So it has a lot of energy. Why? Because it has a ton of carbon and hydrogen bonds, just like we mentioned earlier. Right? It has a lot more carbons and hydrogens. It's a more compact fuel reservoir, so animals can store more energy with less weight, and plants with starch, they're going to also be able to store this as a starch. Um, the next thing here is cushions vital organs. So in your body, some people always think to themselves, I want to have 0% body fat. Well, if you had that, then it would be a really bad day because many of your organs require fats in order to cushion them and protect them. An example is your kidney, also your eyes, right? There's going to be fat deposits behind your eyes to make sure they stay in the same spot. Kidneys are going to have a fatty deposit around them to make sure they stay in the same location and they stay protected, right? So in case you get hit in the back, it's not automatic, just you hitting the kidney. So it's very important for that. Not only that, but insulation, specifically in mammals. We use fat as an insulation so that we can maintain our body temperature. The next type of lipid we can talk about are the phospholipids. Phospholipids are compounds with a few things. They have a glycerol, 2 fatty acids, and 1 phosphate group, which is a big deal. Um, why are they so important? Well, the thing with phospholipids is that they are used for things like the phospholipid bilayer of your cells. So when you're looking at a phospholipid, they have 2 major components. You have the hydrophilic end and the hydrophobic end. Anything with a philic or phile on it means that it likes something. So hydrophilic means that it likes water. Philic means like, hydro means water. Hydro water phobic means water fearing. Hydrophobic. So there's 2 ends. The end that wants to be near water, the one that wants to be away from water. If you take these 2 things and put them together at opposite ends, you can create a bilayer of phospholipids. This is what creates our cells. That's why there's one side of our cells that faces the outside of the cell and one side that faces the inside. It helps separate the components of the cell from the outside world and literally allows life to happen. So it is really, really, really important for us to have. And the next one on here is steroids. All right, so steroids are lipids that have 4 fused carbon rings together, and they are a functional group also known as cholesterol. So cholesterol is an important steroid. We know of them. There are many different types of cholesterols in the body, and it is a type of steroid. All right, steroids are also precursors for many other things. Cholesterols are also precursors for many other things in the body, such as sex hormones and bile acids. So we see these all over the place in our body. And here's a couple of examples of what that looks like. All right. So those were lipids. The next section we're going to cover are going to be our proteins.